Mycotoxins cause, without any doubt, huge economic losses due to their impact on animal health and animal production. The annual losses in US livestock production are around $1.2 billion, and it is estimated that we face a loss of 3 billion euros in crop production across Europe every year. Interestingly, first cases of mycotoxin impacts were already published in 1928. They described hyperestrogenism in swine after being fed moldy feed. It took then 30 years that the fungus Fusarium graminearum was isolated, and it was identified as the main producer of a toxin related to these symptoms. The toxin was then called zearalenone. This is quite a scientific term, and I prefer to keep it simple and use the tech term, which is ZEN. It took then another 30 years that at EU level, the importance of ZEN was recognized. Today, we will discuss both the impact as well as the mitigation of this fungal toxin. A very warm welcome to the world of BIOMIN Science and Solutions, presenting to you our next generation of mycotoxin risk management, called ZENzyme. I am Eva Maria Binder, and I'm responsible for the innovation process in BIOMIN. Today, I have the pleasure to be your host. The benefits of having this online discussion are obvious. We are broadcasting worldwide, and all of you have a front row seat. We would also like to get into a dialogue with you. Therefore, please use the question setting at the right bar of the GoToWebinar platform in order to place your comments and questions. They are all very highly welcome. It is really a serious problem that we are addressing today. You will not only learn about the latest scientific developments, you will also experience the power of ZENzyme, the solution to counteract the mycotoxin ZEN. Before I have the honor as well as the pleasure to introduce today's panel, let's check out together the consequences we have to face if we do not take action against this fungal toxin.
Zearalenone, or ZEN, is a mycotoxin produced by several fungi species. Findings of the BIOMIN Mycotoxin Survey, the longest-running and most comprehensive dataset on mycotoxin occurrence, have revealed that ZEN occurs in all kinds of commodities worldwide, especially in corn, wheat, and soy. Due to the structural similarity to estrogen, zearalenone latches onto the estrogen receptor, which can induce hyperestrogenism.
Aus praktischen Erfahrungen wissen wir, dass Mycotoxine wie zearalenone besonders in der Schweinehaltung große wirtschaftliche Schäden verursachen können. Dabei sind Fruchtbarkeitsstörungen wie erhöhte Umrauschrate und Aborte oder Symptome von Hyperöstrogenismus bei Jungtieren nur einige der Probleme. Bereits eine geringe, aber länger andauernde Belastung mit Mycotoxinen könnte die Gesundheit und damit die Leistungsfähigkeit unserer Tiere beeinträchtigen. Auf unserem Betrieb ist daher ein umfassendes Mycotoxin-Risikomanagement von der Ernte des Getreides über die Lagerung und Probennahme bis zum Einsatz spezieller Futtermittelzusatzstoffe die wichtigste Grundlage für konstant hohe Leistungen.
Quite a challenge, isn't it? Now it's time to introduce our fellow guests. They are all top world scientists in mycotoxin research and mitigation. Thank you all for being here with us at this live event. And most importantly, an information to be shared with the audience. All of us here have been PCR COVID tested just right before this meeting, so we are safe. This is good news. First, I would like to welcome the head of the Institute of Bioanalytics and Agrometabolomics at BOKU's Department IFA Tulln, Professor Rudolf Krischka. You have been repeatedly the most cited person in mycotoxin research, and our collaboration lasts back for more than 25 years. Welcome here, Rudi. Dr. Veronika Nagel, you are a vet and one of our key researchers at BIOMIN. Veronika, you were one of the first scientists to do piglet trials with ZENzyme. I look very much forward to learn about your experience. A very warm welcome to the head of the Department of Poultry Science at the University of Georgia, US. Thank you for joining Professor Todd Applegate. Unfortunately, you couldn't travel to Vienna due to the current pandemic situation, but it's great to have you connecting live to this event from Georgia.
Thank you, Eva. It's great to be here.
Furthermore, I'm very glad to welcome the head of the Institute of Animal Nutrition and Functional Plant Compounds at the University of Veterinary Medicine, Vienna, Professor Cendrim Sebeli. And here with us is also BIOMIN's R&D Program Director, Gudrun Vogtentans. Gudrun, you are in charge of the team that developed the SAN-degrading enzyme. I look very much forward to learn about the journey in this regard from you. And finally, from the Competence Center for Mycotoxin Risk Management at BIOMIN, I gladly welcome Dr. Paula Kowalski. Paula, you're the Global Product Manager for the Mycofix product line, and I would like to invite you to share with us BIOMIN's approach on how to tackle the problem caused by SAN in livestock production.
Thank you so much for the very nice introduction, Eva. So BIOMIN has been working now for the past almost 4 decades in providing the most cutting-edge solutions for the mitigation of mycotoxins in animal feed. And today, without a doubt, we are world leaders in mycotoxin risk management. Now, the absorption of zearalenone is very limited, and this is due to its chemical structure. So if we compare it to absorbable mycotoxins like aflatoxins, conventional binders such as yeast-based products are less effective. The absorption of zearalenone with these kinds of binders is only around 20%.
As we know that absorption is obviously not the right approach to tackle SAN, what would be the alternative?
So the best strategy to counteract non-adsorbable mycotoxins, as the case for zearalenone, is the use of biotransformation. And this is using patented products with a proven efficacy. And this is why we are here today, and I'm very happy to be able to be with everyone here, because we are launching the latest technology or the latest development for the zearalenone mitigation called ZENzyme. The reaction is very fast, it's targeted, and it's irreversible. And the resulting metabolites are no longer estrogenic and no longer harmful to the animal. Now, back in 2004, BIOMIN began to analyze mycotoxins in all kinds of different commodities and finished feed samples from many different countries worldwide. So I think we cover around 100 different countries. Today, the BIOMIN mycotoxin survey is the longest running and most comprehensive data set on mycotoxin occurrence.
Well, we have been in this now since 2004. How many analyses did we do, actually?
We've analyzed 120,000 samples and conducted over 500,000 analyses. And this is what makes the mycotoxin survey very special. We are able to see very detailed information about the mycotoxin occurrence in these different raw materials as well as finished feed samples. And what we have learned out of the survey is that zearalenone is a mycotoxin that very commonly occurs. And also, and this is the most important part, it doesn't occur alone, it co-occurs with other mycotoxins such as trichothecenes, most importantly deoxynivalenol. And we also see that it co-occurs with fumonisins. Now this co-occurrence is the rule, not the exception, and it's very important because we see that this co-occurrence of mycotoxins can lead to synergism. One of the highlights of the mycotoxin survey is the Spectrum 380, which is based on the leading technology in mycotoxin analysis using mass spectrometry.
So we couldn't have this done alone. We rely on the collaboration and the innovative methods developed by our collaboration partners, in this case at the Hotspot for Mycotoxin Research in Tulln. Rudi, it's really great to have you here. And when it comes to SAN, what's your opinion on further and future methodologies for SAN analysis and also prediction?
Yeah, well, Eva, first of all, as a passionate scientist, and particularly passionate chemist, I have to point out right at the beginning that zearalenone is not only an estrogenic mycotoxin, it is also a fluorescent molecule. So this fluorescent actually, when being exposed to UV light, this estrogenic mycotoxin exhibits is utilized by many laboratories around the world. So that is why liquid chromatography coupled to fluorescence detectors is the major method used around the world by many labs. Having said that, there is based on the recently published literature, and that is true for the last decade or so. Tandem mass spectrometry subsequent to liquid chromatography is the method of choice when it comes down to measuring at the most sensitive level, also when we need to provide ideally also the identity of the molecule, and if we have the task to measure co-occurrence mycotoxins. So not only focusing on zearalenone, but considering zearalenone as one of many, though being very important, co-occurring mycotoxins and other secondary metabolites being produced by plants and fungi. So this is a fact which we have been facing for many decades, and we are aware of this fact for many decades, that we have co-occurrence of mycotoxins and also frequent occurrence of these secondary toxic metabolites. And that is something which the FAO, the Food and Agriculture Organization, has pointed out some 30 years ago, that about 25% of the agricultural crops are contaminated with mycotoxins, and this figure has just been recently updated, and this latest study clearly testified to the fact that it is a clear underestimation of this FAO figure, and in fact there are some 80% of the agricultural crops which can be considered as contaminated with mycotoxins at the, above the limit of detection. And this co-occurrence on the one hand of mycotoxins, and particularly this upgrade or update where we see that we have some 80% of the crops being contaminated, is particularly true for zearalenone, and we have an occurrence data, and Paula has pointed out that there has been this BIOMIN survey study where also our university has been involved, that this are data which shall not only be taken serious when we are to provide a full risk assessment of the feed in food, but also shows that we need a methodology which ideally covers the entire spectrum of these toxic secondary metabolites and other co-occurring mycotoxins. So this is one end of the spectrum, so to say, the most sophisticated but also costly methodology, which is ideally to cover the entire spectrum. But certainly there's also still a need for rapid methodology, utilizing especially immunoassays, which is still the most dominant methodology when it comes down to the rapid and less costly determination. Just in the last few years, there have been a couple of components which have been added, innovative components to these immunoassays, such as magnetic nanotubes or zearalenone-specific aptamers. Still with the limitation that rapid methods in general, though being innovative, have limitations when it comes down to measure more than one mycotoxins, more than just zearalenone. That's a challenge. It's also a challenge from a sensitivity point of view, and that is particularly true because of matrix effects. So the more complex, the more complicated a matrix is in the food and feed arena, the more we have the demands to involve chromatography, and particularly by mass spectrometric detection.
Thank you, Rudi. You explained now that ZEN is really a huge problem in worldwide crop production. But what about its effect on livestock industry? Let's have a look at this.
The negative effects of the mycotoxin zearalenone is incredibly important for swine farms, especially sow farms and breeder farms.
This mycotoxin is responsible for vulvovaginitis and estrogenic syndrome, not only in reproductively active swine, but also in prepubertal gilts and even piglets.
Zearalenone mainly causes estrogenic effects in pigs. In pregnant sows, it increases the occurrence of abortions and stillbirth.
Another thing what we also see in swine production is that the piglets has a larger vulva, red and larger vulva, which is a sign of of a high level of zearalenone.
This toxin can bind with estrogen receptors in cells and gills, resulting in negative impacts on the reproductive organs.
We have financial losses due to the cell elimination by abortings, prolapse, and others. Zen control is essential to avoid reproductive and financial losses.
Veronica, what are the major effects of Zen in swine from a vet's perspective?
Yeah, so pigs are especially sensitive to zearalenone. This is because after intake, this mycotoxin is converted to an even more toxic form in the body. The first and the major target of zearalenone in pigs is the reproductive tract, where it causes functional and morphological alterations. Clinical signs, for example in prepubertal gilts, include enlargement of the reproductive tract, and this is most easily visible by reddening and swelling of the vulva. Later on in sows, zearalenone can impair the whole reproductive performance, for example by decreasing the quality of oocytes, prolonging the weaning to estrus intervals, causing pseudopregnancies, or as well a higher rate of embryonic mortality.
So Zen affects not only the sow, but also the offspring, right?
Yeah, indeed. Zen exposure in sows subsequently affects the health of the newborns. So here we see a higher percentage of weak-born piglets with spray legs or trembling, and also signs of hyperestrogenism. Unfortunately, the males are not protected from this mycotoxin. So in boars, we have decreased quality of sperm or reduced libido. And besides all those effects on the reproductive tract, Zen potentially compromises liver metabolism, gut health, as well as the immune system.
There is no doubt now that swine are highly sensitive to Zen, but what about other species like, for example, poultry?
The introduction of a pure enzyme for the biotransformation of zearalenone is a major step forward in mycotoxin control. Unfortunately, currently, there is a trend for an increase in mycotoxin contamination, including zearalenone and its masked forms in feed raw materials today.
Zearalenone is a significant effector of the breeder reproductive system and functions. It affects egg quality, increases embryonic mortality, leading to reduced hatchability. Furthermore, it also affects feathering of birds by compromising methane utilization.
Zearalenone can cause polycystic ovary syndrome. So this is because of the excessive of estrogen.
Continuous consumption of zearalenone contaminated feed could lead to detectable amount of zearalenone and or its metabolites in the blood, in the liver, intestine, content, urine and feces.
The impact of zearalenone on breeding flock reproductive performance is very obvious. In broiler breeding industry, nobody wants eggs if they are infertile. And since we're always seeking opportunity to improve fertility, zearalenone control in broiler breeder flocks is extremely important.
Todd, what is your scientific opinion and evaluation on the effects of Zen in poultry?
So when we think of Zen in the bird is very rapidly absorbed, and it's rapidly transformed into its toxic metabolites, right? So if we think of then the toxic effects of Zen in the bird, realize that Zen by its molecule, its shape is very similar to that of estrogen. So a lot of the occurring effects tend to be much more reproductive. So in the bird, those concentrations of estrogen circulating are much higher, and therefore the bird has been thought in the past to not be as sensitive as some of the mammalian counterparts, such as the pig, which is very sensitive. But we still see these long-term chronic exposures to zearalenone are having some long-term chronic effects on the bird itself. So as far as susceptibility, the turkey is very susceptible to these chronic exposures, followed by broiler breeders, then layers, ducks, and lastly, broilers. So with chronic exposure, especially during that pullet phase of rearing, we're seeing delays in reproductive tract development. Those birds may not peak as well in production and may not have persistency of lay over the long term. And part of those laying effects that we see are due to cysts on the oviduct itself. inflammation to the entire reproductive tract. And probably the most visible symptom that we see is that of a reduced eggshell quality in those birds that have been chronically exposed to zearalenone. The other thing I'd like to mention, just when you're looking for concentrations of zearalenone, they tend not to occur by themselves, and it is in the top 3 for frequency of co-occurrence with other mycotoxins. So we're expecting some of those synergistic effects of that co-occurrence overall on the animal to lower overall performance.
Ladies and gentlemen, we received a lot of information so far. Please post your questions. It's really a pleasure for all experts in this panel to answer them. So far, we have seen that swine and poultry are clearly affected by this mycotoxin. But what about ruminants and dairy?
Zearalenone is detrimental to ruminants as it reduces reproductive efficiency. Poor reproduction can result in fewer calves being born. Calving initiates milk production, which is essential to dairy farms, while beef calves are raised for meat production.
Zearalenone kann den Hormonhaushalt von Milchkühen wie folgt beeinflussen. Erstens durch frühzeitige Eutervergrößerung, zweitens Scheinbrunst, welche die Erkennung der echten Brunst schwieriger macht, und drittens nicht erfolgreiche Besamung bzw. Aborte und damit verbundene ökonomische Einbußen.
ZEA can cause swollen vulvas, uterus prolapse, ovary cyst, and abortion. Therefore, negatively influence the reproduction level of the dairy cows.
Dairy cows, ruminants in general, especially the dairy cows, but also dairy ruminants, but also the young, especially the young heifers, for example. In general, the breeding animals are not excluded by the effects of the ZEN, and this is mainly due to the reason that by the degradation of the ZEN in the rumen, we do not have detoxification, as the case for many other mycotoxins, for example. Rather, we have an upgrade of the toxicity, and this is unfortunately due to the metabolites that are being released in the downstream degradation in the rumen, which are mainly the alpha type, for example, of the zearalenone, the so-called alpha zearalenone, which there are many studies indicating that the estrogenicity is much higher than the mother molecule. And in conclusion, of course, the consequences for the heifers, but also for dairy cows, are manifolds. We have problems with detection of the real estrus, for example, but there are also disruption of the whole fertility parameter, the fertility traits in the cattle as well, and at the end of the day it leads to the disruption of the fertility parameters, and you have a decreased fertility performance of the cows, which leads to increased or enlarged calving interval, which is one of the main reasons of decreased fertility parameters and reproductive parameters in dairy farms nowadays, no matter if you look at here in Europe, but also in other countries as well.
So summarizing, we can say that ZEN is present worldwide. It causes serious reproductive as well as health problems in animals, and it leads to massive economic losses. So we wouldn't be here today if we could not share with you a solution. I think you got some for us, right?
Yes, exactly. So despite all of these problems, there is a solution, and this is our latest development in zearalenone mitigation. ZENzyme is the first and only purified enzyme that is capable of degrading zearalenone to non-estrogenic and non-toxic metabolites. And the reaction is, it's immediate, it's very fast, and most importantly, it's irreversible.
Therefore, biotransformation is the best strategy to counteract non-adsorbable mycotoxins using patented components. They are effective and EU approved. This makes ZENzyme the state-of-the-art technology in zearalenone degradation. The reaction is specific, irreversible, and immediate. The results are non-estrogenic as well as non-toxic molecules.
We have already learned now that there is a very much needed solution. Now we would also like to know more about the scientific background. Gudrun, could you tell us a bit more about the story of the discovery and the development of this enzyme?
Yes, thanks, Eva. I would like to start out with acknowledging and thanking the whole ZENzyme team and everybody who contributed to this success. It is an exciting moment for all of us because we are bringing a product to the market utilizing really the most advanced technology, zearalenone detoxification technology using enzymes. So now to your question, how did we discover ZENzyme? So we started out screening soil samples from different cornfields, and we were screening for activity on zearalenone, offering zearalenone as only energy source. On the active samples, we did single colony screening, isolation, in order to identify the strain. And then in the next step, where we were screening for the most suitable strains, the most active strains, it was really important to have the LC-MS method available, because with this method, we could identify the strains that were highly active, looking at the zearalenone degradation profile, and on the other hand, we could identify the samples that resulted in non-toxic, non-estrogenic products. The next step was then to clone the genes and to express and isolate the enzyme. So with that, we could really verify in vitro enzyme activity. To turn this finding now into a product, we needed to verify the enzyme performance in vivo. So the efficacy of ZENzyme was verified in vivo using biomarkers. In every single feeding trial, we have seen a significant zearalenone reduction, which was due to the activity of the enzyme. So clearly in this process, we were able to identify an enzyme that can degrade zearalenone. It acts on it fast, giving us non-toxic products. And most importantly, or similar important, it is that this enzyme is also active in vivo, working in the animal.
So in a nutshell, it's just a very simple, sophisticated approach with high-level expertise, right? Yes. Now, in the meantime, we received a number of questions from our viewers. And the first one, Paula, you might like, because it's directed to you. Somebody's asking, by when will this product be available?
So ZENzyme is part of the Mycofix Plus 5-point set, and it will be available straight away in the countries where the registration process has been finalized. You just need to contact your BIOMIN representative to make your order. Mycofix Plus 5-point set can be added directly to the feed during mixing. It is pellet stable under standard pelletizing conditions, and it is recommended at a dose of 0.5 to 2 kg per ton of feed.
Can you give us some more information about the actual effects and also the advantages of this product?
The product has numerous benefits. So first, as Gudrun mentioned, the activity of ZENzyme is extremely high. It's very fast in degrading zearalenone, which of course is important to enhance the reproductive performance of the animal. We are able to combat a very broad spectrum of mycotoxins using this product because Mycofix Plus 5-point set not only targets zearalenone using ZENzyme, but we are also able to counteract fumonisins, trichothecenes such as deoxynivalenol, ochratoxin A, aflatoxins, ergot alkaloids, and other emerging mycotoxins, as well as endotoxins. So it's very complete.
So you can make a lot of friends with just one gift?
Exactly. And as all BIOMIN products, we have a We have product ingredients that are patented and with proven efficacy and safety, and also backed by the only EU authorized by the only EU authorizations, which of course shows the high quality of the product. We have an enhanced bioprotection, which is very important when the immune system of the animal is weakened, and also the performance of the animal is not so good due to the presence of these mycotoxins as well as endotoxins. And lastly, we have a verified economic advantage. So this is important to protect your profits in the case that you have performance, health, or reproductive issues due to the presence of these mycotoxins.
Now, the next question is directed to Todd. Todd, one of our viewers asks how Mycofix Plus 5.Z can relieve the economic impact caused by the negative effects of ZEN in poultry production.
So usually we'd have to back calculate to overall production. So if we look at egg production in particular, we see that on average across studies that typical concentrations of ZEN over a long term are going to reduce egg production by about 6%. So if we're thinking of those dollars as a laying hen over the cycle of production, that may be up to $6 to $7, just depending upon where markets are. If we're talking breeding birds, that valuation of not having a viable chick would then be much higher. So encompassing a lot of the overall effects, we need to not only think of the effects on the female for degradation of fertility, but then also that of the male. So anything that we could do to alleviate that overall toxicity to the animal, we'd experience improved fertility to both the male and the female. For females, we would expect overall improved fertility, improved hatchability. When we're talking about shell eggs, we'd be talking about sustained eggshell quality for those eggs going through the entire system to be able to reach the customer, and overall improved reproductive performance, which will ultimately lead to improved profitability. The biggest impacts I see are over the chronic exposure that the animal would be experiencing. So over those chronic exposures, if you have that over the length of a laying cycle, roughly that'd be about 34 eggs for the bird that you'd be able to then to recover.
Thank you, Todd. Actually, Veronica, this brings us to the next question. How can we measure the efficacy of such a product in pig? The term biomarker was also mentioned in one of the questions.
Yeah, precisely, Eva. So we scientists, we truly love these mycotoxin biomarkers because under controlled experimental conditions, they're simply the best tool to show the efficacy of a product. They enable us to measure the biotransformation of the toxin and therefore the specific mode of action of the enzyme. Also, they are a strict requirement in many registration processes worldwide, particularly the European Union. In case of ZENzyme, Gudrun gave already a small hint. So here we are able to measure the non-toxic metabolite, the hydrolyzed zearalenone, in pigs, in feces. So when we administer ZENzyme, the levels of these non-toxic metabolites increase in feces, whereas the one of zearalenone decrease. And thereby we can nicely show that the product is properly working inside the animal.
Gendrim, animal welfare will certainly reshape our industry. What could be the impact of ZEN in this regard?
Yes, of course. This is the welfare. I mean, the question is that the welfare nowadays, the awareness towards welfare is being increasing, and this is right. So the welfare from the perspective of consumers, as a consumer, for example, you expect to have milk or milk products or meat and other animal products from healthy animals, from animals that feel well, animals that are fed well, but also animals that are reproductive. Without a reproductive animal, you cannot have milk at the end of the day. And so that the pain, but also the distress that are caused due to the contaminants and then playing a role when it comes to the dairy cow, of course they affect the welfare of the dairy cow. And on that, saying that, the expectations are that the ZENzyme having the activity to enhance the degradation of the ZEN in the rumen, something that we have, for example, also observed in one study that we just published a few weeks ago, where we observed that the ZENzyme accelerated the degradation of the ZEN and especially minimized or lowered the concentration of the highly estrogenic alpha cell, for example. And based on these results, what you expect from the point of view of the welfare, but also reproductive performance on the cow or the dairy heifer, are that the performance, the reproductive performance will be improved, so that there will be no reasons or no consequences of the distress that might be caused to the animals due to the presence of those estrogenic mycotoxins, but also the performance of the animals will be enhanced and be improved in general, especially the reproductive performance. And actually, the decreased reproductive performance is one of the key parameters that affects not only the welfare of the animals, but also the profitability of the dairy industry in general. So to conclude is that the outcome is expected to be manifold in dairy cows.
Good one. We got one question with regards how and where is ZENzyme working exactly?
Yes, so I would like to start with the how. ZENzyme hydrolyzes zearalenone into H-zen. And this, we developed the enzyme for high affinity and high specific activity towards zearalenone. So this reaction is very fast. We determined the kinetic parameters, including the turnover rate. We also know that ZENzyme can work at very low substrate or zearalenone concentrations. What we have seen in trials is that as soon as there is moisture available, ZENzyme starts working. So we need around 20% humidity. This means for in the animal, so as soon as the animal ingests feed containing the enzyme, the rapid detoxification starts. So we see high enzyme activity in the saliva fluid, but also we were able to verify the ZENzyme being active in the gastrointestinal tract. So now looking at all the different species, I think Veronica already explained the efficacy of ZENzyme in pigs, and Professor Sivili explained the benefit of ZENzyme in lactating cows. We could also verify the positive or the efficacy in rumen, and for crop we could verify that the enzyme and for poultry, we could verify that the enzyme is active in crop. So all our trials, all our data points towards that the reaction happens early on. So the detoxification happens early on. And this really minimizes the negative effect of zearalenone, giving the full benefit to the animal.
Thank you, Gudrun. Now we touched questions regarding animal welfare, economic losses. What also came up for sustainability. Do we have any impact with this new product generation on sustainability? Who would like to take this question?
If it's okay, I can take it.
Thank you.
So I can speak about pigs, where we've seen in numerous trials that ZENzyme is indeed a unique solution counteracting reproductive problems caused by zearalenone exposure. So the application of ZENzyme results, for example, in a decrease of the pathological changes in the reproductive tract. It increases sperm quality and libido in pigs. in boars specifically. And in sows, we have a higher number of piglets per sow and improved farrowing rates. If we consider now that one non-reproductive day per sow can cost up to €2, it's clear that ZEN can have a marked impact on the economics. So by increasing the reproductive cycles per sow and by avoiding return to heat, we can have a more sustainable animal production.
Thank you. Pretty clear to me. Ruli, now you published a paper on ZEN nearly 30 years ago. What's now your opinion on the future of ZEN analysis and particularly on prediction of ZEN occurrence?
Well, first of all, thanks Eva for reminding me of my age. It is indeed amazing, if I may say so, that my team and I at BOKU IFA have maintained an interest for mycotoxin research for now almost 30 years, and more than 2 decades have also been in collaboration with you, which makes me very happy as well. Digging a little bit more in the history, let me also point out that I had the pleasure to be part of the FAO-WHO conference, which took place in 1998 in Tunis. And believe it or not, the conclusion back then was in relation to zearalenone, that there is no effective method available which destroys zearalenone, and actually that indeed FAO demanded for a worldwide research effort to come up with methods which can inactivate zearalenone. So with this historic angle, I think it's particularly amazing to see these latest developments which have been discussed today. And to accommodate your question a little bit more on the spot on prediction tools, I think these are tools besides the analysis which is carried out in labs, which becomes increasingly important to ensure foods and feed safety and feed security at both ends. It is something which has to be pointed out that zearalenone is rather a pre-harvest issue. So as a result of Fusarium graminearum and Fusarium culmorum mainly infecting maize, this is an issue which occurs mainly at the pre-harvest level. So zearalenone is found mainly at this stage. Having said that, it is also a fact that if the grains are not stored properly under dry conditions, this issue can easily transform also into a post-harvest issue. And in both areas, in pre- and post-harvest, there have been a number of prediction tools, so-called decision support systems, which have been developed, particularly over the last few years. So in the pre-harvest arena, we have models where, for instance, the type of soil, the genotype of the crop, historic weather data, actual weather data, but also the life cycle of the fungus is taken into consideration to build up models which help you actually, based on the actual weather data, which are fed into the system for the farmer to provide them with an information on the risk that a certain mold and a certain mycotoxin occurs at the pre-harvest level, and then certain measures can be taken, as for instance spraying fungicides at the right time or early harvesting, etc. On the post-harvest side, and that is something which is particularly important in some areas of the world, particularly in China, where some amazingly huge numbers and actually amounts of grains are stored in silos. So each of these buildings containing some 6,000 tons of grains. And we have, in the project which I had the pleasure to coordinate, my toolbox, in collaboration with several partners, installed sensors there which measure temperature, relative humidity, but also, in most importantly, carbon dioxide as the most early and most rapid indicator for mold growth. And actually, by linking this physical data to biological models, we are able to come up again with a risk indication whether or not mold formation, zearalenone uptake can be recognized or can be expected in the silo. And then again, the owner of the silo, the farmers, can make a decision whether or not to spray a fungicide to aerate or to remove, for instance, the kernels. And these prediction tools, Eva, which you have pointed out, is something which becomes increasingly important in view of climate change, global warming. So extreme weather conditions result actually in increased levels of mycotoxins, particularly unexpected occurrence. So we have an unexpected, unpredictable levels of mycotoxins, and particularly of mycotoxin patterns. And this is why prediction tools, as described in the last few minutes, are becoming increasingly important in combination with the analysis which is carried out in the lab in order to tackle the mycotoxin control, the mycotoxin issue, and also to control this very important topic for food and feed safety.
Thank you, Rudy. Based on the available data, we assume that climate change will affect ZEN occurrence worldwide. Now, how does the climate change affect animal production? Veronica, what's your view on this?
So climate change will definitely affect animal production. Actually, it's already doing so, because we know that heat stress is one of the major reasons for seasonal infertility in swine. So during warm summer months, we have higher incidence of reproductive problems, such as reduced farrowing rates, reduced milk production, or prolonged weaning to estrus intervals. It's a bit tricky to put this into numbers, but there are estimates that in the US swine industry alone, heat stress causes economic losses of up to $300 million per year. For the next years and decades, it has been projected that heat waves will not only occur more frequently, but they will also last longer. So in this regard, I think we are definitely facing challenging times ahead of us, and we should give our best to maintain animal welfare and productivity.
Thank you all. We actually received a huge number of questions, but unfortunately we cannot answer all of them right here. Please be assured that we will provide complete FAQs. Additional information about ZENzyme and Mycofix Plus 5.Z can be found at the link zenzyme.biomin.net. And we also compiled all the information we shared with you today in the zearalenone compendium. Not only the information we shared today, even more. Be surprised. Thank you for your inputs and the discussion we had. We met now world-leading scientists with regards to mycotoxin research and mitigation, and it was really a pleasure having you here and most enjoyable to discuss with you. It is absolutely obvious, ladies and gentlemen, for me that today's session was just the beginning of a common journey. Thank you so much for being our fellow travelers. It was great to stay in touch with you in this way, and please take good care and stay healthy.